Hey, how's it going? Today we're going to be talking about genetics and Punnett squares. So the objectives for today's lecture is to know the major vocabulary so that when we talk about Punnett squares and genetics, you know what to do. Some tips for how to do Punnett squares and how to predict some ratios with Punnett squares. So just a little highlight, we're going to talk about what a gene is. A gene is a section of DNA that codes for protein. A section of DNA that codes for protein, and this is directly related to the central dogma. So if we took a look at DNA, we could find a gene in different parts. So if this is our DNA, we might have the gene for height, we might have the gene for earlobes in two different spots, but they're specifically on a section of the DNA, and it codes for a specific protein that affects your appearance or affects in a disease state. So when we're looking at these genes, we can describe different things about a person. So the first thing we can describe is your genotype. Your genotype is essentially your genes. What are the codes on your genes that you have? What are the traits that you have? And these traits can be categorized into two things. Dominant, which is usually represented by a capital letter. And the reason why it's called dominant, because if you have one copy, this is going to show up as your phenotype. So some examples of dominant traits would include things like brown hair color, brown eyes, and other traits like that. You'll see a lot of this when we do our human traits lab. You'll see which traits are dominant and recessive. So the opposite of dominant is recessive, and these are represented usually by lowercase letters. And in order to show a recessive trait, you need to have two copies. So some examples of recessive traits mean that you need to have two copies. Examples of this would be things like sickle cell and hemophilia, where you would need two copies for this to show up. So again, these are your genotypes. They're genes that code for a trait. They're usually represented by letters. Some examples of how a genotype might show up, be it capital A or lowercase a. Capital A, or lowercase a would be some examples of genotypes. So now that you have your genotype, we can also describe yourself with a phenotype. The phenotype would be the physical traits that makes you who you are. So some examples of that, for myself, that would be brown hair, brown eyes, and short. These could also include things like hitchhiker's thumb, and any other trait that might come to mind about earlobes, hair on the middle digit would all be examples of phenotype. And the way that that works is your genes, or your genotype that we just talked about, affect your phenotype or physical characteristics through the central dogma. So because of your genes, it affects who you're going to be, what you're going to look like. So now we can divide those into two things, heterozygous and homozygous. Underneath heterozygous, you basically have one option. You have one dominant allele, or one dominant gene, and one recessive copy. So this would be your capital A, lowercase a. This would be your genotype, but you would have a dominant phenotype. In contrast, if you're homozygous, there's kind of two options you could go down. You could be homozygous dominant, meaning you would have capital A, capital B, and you would look just like a heterozygous. You would have the dominant phenotype, hence that's what dominant means. In contrast, if you were homozygous recessive, you would have the lowercase letters, little a, little b type things, and you would have the recessive phenotype. So these would be things like blue eyes. You would have a recessive phenotype because you have two copies of the alleles. So now that you have this information, what can you do with it? You can use something called a Punnett square. So a Punnett square is a tool came up with by Mr. Punnett when dealing with genetics in order to answer some simple questions about what you think the offspring would look like. So they came up with these questions on what kids look like, and what they did was they were able to take the parents' traits, and we're looking at a one-trait cross, and we're able to come up with the gametes. So gametes against sex cells. If you took a cell, you could have a big A and a little a. If you think back to meiosis, we're going to separate on two sides of the plane. You have an equal chance of getting a big A or a little a. With the lowercase letters, you just have an equal chance of getting either a little a or a little a. So if we cross these kids, we'll put our Punnett squares inside, you had a big A, a, little a, little a, little a, big A, little a, little a, little a. So what does this mean? This means that we have, for phenotype, we have 2 dominants, 2 recessive. In our genotypes, we have 2 big A, little a, 2 little a, little a. So to recap, what did we do for the steps? We found the gametes. We made the square. And third, we summarized our results. Some other things you might want to do is to make a key. In this case, we didn't set up what the trait was, but if we knew that capital A meant brown hair and little a meant blonde hair, we could make a key, and that way it'd be easier to find out what the dominant and recessives were. So let's try another one, going through those same steps. Step 1, we'll look at what the parents are. We'll set up a key. We'll say, for example, big T, we'll say is a long tail, is dominant. And a little t, we'll say it's short tail. We're looking at cats, mice, some animal with a tail. And we have one parent, we'll say the mom is big T, big T. She has a long tail. And we'll say the dad is little t, little t. So we can set up the possible traits, put them on the outside. On the outside, we'll put the parents, and then we'll put the inside. Go across and fill in. We'll find out that 100%, all 4 of the parents are going to be, all 4, despite the father having little t, all 4 of the offspring are going to have long tails. And all 4 of them are going to be heterozygotes with a big T and a little t. It's another example of a Punnett square. So the next thing I want to talk about was a test cross. Test crosses are used to determine unknown genotypes. So like I said earlier, when you had someone with a dominant phenotype, they could either be big T, little t, or they could be big T, big T. So how do you know which one they are? You could mate them with somebody who's recessive, homozygous recessive, and you could find out based on what their offspring look like. So go ahead and take a moment and try out these Punnett squares. What would it look like for a homozygous dominant to mate with a homozygous recessive, or heterozygous to mate with a homozygous recessive? So take a moment, go ahead and try that. Okay, so hopefully you tried it. I'm going to work through it and let you see what that might look like. So again, we're going to put the recessive traits on one side, and we're going to pair another parent on top. So if we do this, we'll find that the ratios are different. In the homozygous example, all the offspring are going to look like the parent. So we have 100% capital T. In this case, we'll have 50% capital T, 50% lowercase t in terms of phenotype. And that's how test cross works. You can find out if something is considered true breeding, meaning that they're capital T, something that's true breeding. And this is especially useful in genetics when looking at breeding, when trying to do genetic counseling, when trying to find out if a disease would get passed on to the offspring. So what happens when you do a 2-trait cross? You have to do actually a little bit of math. So you might know how to get the gametes by using a process called FOIL. FOIL stands for first, outside, inside, last. So to get all the possible gamete combinations, we want to look at all the combinations of A's and B's. So we're looking at 2 traits, for example, hair color and eye color. In peas, this could be things like flower color and height of the pea plant, things like a wrinkly shell or broadleaf or skinny leaf. And so what we'll do is we'll do the same thing of first, outside, inside, last. So our first combination, big A, big B. That's one of the gametes, one of the types of things that show up in the sperm or egg. We'll go ahead and do the outside. So we have the outside. So our option is big A, little b. And if we go through the top, we have inside. So that would be a little a, big B. And lastly, our last option is the last. So that would be little a, little b. These are the 4 types of gametes that this organism can provide. So somebody who is heterozygous for 2 traits could create 4 different type of gametes. What could you then do? There are 2 ways to then make this Punnett square. So let's say we wanted to mate someone like this. You could use a big Punnett square like this. Again, we use FOIL to come up with all the possible combinations so that you would end up having things like first, outside, inside, last. And then we do this for both traits, both parents. And you could do the Punnett square this way. For example, you'd find out 1/16 would be dominant-dominant, 1/16 would be recessive-recessive, and you'd have a blend in between. I'll let you go and continue that. You'll end up finding ratios of things like 9:3 to 3:1. And in terms of phenotypic ratios, you'll end up finding 9 dominant, 3 dominant recessive, 3 recessive dominant, and 1 recessive recessive. If you continue through the Punnett square. So go ahead and try that out. If you have any questions about it, please ask. You can also get the ratio using the rule of multiplication. So the rule of multiplication says that for any condition you have and, you would add together. So some years we do a lab on this. We'll do the rule of addition, the rule of multiplication. We'll talk about how these things work mathematically. But the idea is you could separate the traits. So we'll put A's on one side, B's on another. And if you wanted this trait, this is a 1 in 4 chance. This trait is a 1 in 4 chance, where we want the individual to be capital A, and capital B. You'll end up with 1/16. Go back to the previous slide, you'll find that the odds for these two are the same. If we also do that for these traits, you'll end up with the same thing. The ratios for these two will also vary a little bit based on what you want, but the idea is that you could have the rule of multiplication to create and a condition where you would use both the rule of multiplication to find things out. So in summary, you find that there are some important words you should know for your genetics vocab, things like heterozygous, homozygous, and test graphs. And we talked a little bit about how to do Punnett squares. And I wanted to just remind you, the biggest tip for Punnett squares is just to practice. Make up problems, come up with things, talk about things, fish, chickens, look up traits. The easiest thing is to just practice, because really practice makes perfect, and you get a lot better by just doing them. So what are we going to talk about next time? We'll talk about some complex genetics, things that are a little bit more complicated than these simple Punnett squares. We'll talk about things like blood type, eye color, diseases, and sex-linked traits. So keep that in mind for next time for all your crazy questions that you're like, I don't really care about peas. This would be more kind of your human genetics. So stay tuned for next time. I look forward to seeing you, and take care. I'll see you in class. Remember, if you have any questions, please text, call, school loop, message. I can't help you if you don't help yourself. So take care and have a great evening.